Hi, this is Dr. Jabali, and in this video we're gonna do topic 2.8, which is cell respiration. Here are the things that we're gonna target. First, we're gonna understand about cell respiration and how it's basically release of the energy from the organic compound. In this case, it's actually glucose, which in the end we're gonna produce ATP. We're gonna learn also that ATP, this is actually the energy, the source of energy of all the cells in our body. We're also going to talk about anaerobic because there is a moment in your life where you can have oxygen and sometimes you're not breathing oxygen. So how does that affect glucose and how does that affect the energy that is coming out of it? Such as in the case here, right? Aerobic, which means it does require oxygen, and how much energy does it come out of that glucose? We're also going to talk about how there is anaerobic happening in yeast and how that's gives the production of ethanol, carbon dioxide, depending on what kind of yeast or bacteria that we're using. We're also going to talk about anaerobic can actually happen in humans and how that produces lactate. And that means basically when your body's starting to get fatigued, there's more lactate acid in there. We're going to talk more about this later on. And we're going to talk about experiments that are done to analyze basically how respiration happens in certain animals or seeds or anything else using the respirometers. So we're going to talk about that experiment because this can fall easily on the test. So first, let's learn the equation of cell respiration. Now, we have to understand that cell respiration, just the word respiration right here, this is basically saying oxygen. That means somehow the cell is breathing that oxygen. So it has to do, if you have trillions of cells in your body, that means every single cell of your body is going through cell respiration. And it's actually going every second. Like right now, when I'm sitting here and talking, I'm going through cell respiration because at this moment when I'm releasing CO2 out of my mouth and I'm breathing oxygen, that means all my cells inside my body are doing cell respiration. Now, what is the purpose of the cell respiration? The purpose is basically to supply energy. Right? Because every cell in your body needs energy to function. It needs energy to do chemical reaction. It needs energy to transport things. It needs energy to open channels. So you need to do cell respiration every second. Now, since the cell needs that, he needs to have something, a source that causes that. And that's where it is. Glucose is that source. See, we eat in order to get the energy from the food, right? And since we're eating that food, we're breaking that food. And when we break that food, energy will come out. So that's what cell respiration is really about. Cell respiration is basically the breakdown of glucose. Basically food, right? So every time we're doing exercise, every time we're talking, anytime that we're using energy, we are breaking down the food inside our body. That's why we eat food in order to keep that storage of energy. And every time we're using energy, we're breaking that food to get it. Now, we also know that in the cell respiration, oxygen is needed. Now, we say that we breathe oxygen in order to be alive, but there's more into the oxygen. Oxygen is needed in order to break down glucose. If you do not have enough oxygen in your body, you're barely going to be able to make any energy out of glucose, and that means you're going to get tired faster. So oxygen is a very important thing in cell respiration. Now, while you are using these two, eventually what something is product, right? What comes out of it. So basically every time you break down food or every time you're using energy, you are releasing carbon dioxide, of course, some water, and of course, the energy that is coming from the glucose. Now, when I say the energy that is coming from glucose, I'm talking about ATP, because ATP, it is the energy that we understand in the human body, okay? So let's talk about this ATP for you to understand a little bit more. See, ATP, as you know, is adenosine triphosphate. Do you have to know to write this full name? No, okay? You can always say adenosine triphosphate as ATP. So, why is it ATP? You can see from the word tri here, which means 3, and phosphate, there's 3 phosphates. So here you have that 1, 2, and 3. You have 3 phosphates. So the word ATP is just means that you have 3 phosphates attached to that adenine right there, and the ribose, of course. Now, this is known as a full battery. Think of a full battery, right? And that means it's full energy. Now, where is that energy found? If you look at this picture, you will notice that the energy is found in the bonds between the phosphates. So right here, there is energy in this bond, and there's energy in this bond, and there's also energy in the bond. So when you break a bond between two phosphates, like if you take this bond out, energy is going to be released. And that energy is the energy that's going to be picked up by the cell to do function. So you can see here in the picture, for example, they just separated the phosphate from the rest, and what came out of it was energy. And this free energy is the energy that you use to do chemical reactions, to make your muscles move, to make channels open, that's the energy. So what does the ATP does? It's just carry that energy that is necessary. And every time you want to take an energy out of it, you just take a phosphate. Now, when you take a phosphate, since there's only two here left, you can see that the word changed to 2. Now there's 2 phosphate, right? So that means it's ADP. So depending on the amount of phosphates, you can know how much energy this compound has, right? This molecule. So here to make a little bit of a summary, what I'm trying to say here is this, is that if you have ATP, right? This means you have attachment of 3 phosphates, and that means you have a full battery, like full energy going on. If you have ADP, which D means 2 phosphates only, that means you can say it has a half a battery, right? And if you have AMP, which M means mono, which means 1, so there's only 1 phosphate, this means a completely very low battery, and that means it's not carrying much energy. So, and where is this energy being carried? Well, this energy is a highlight here in yellow. It is here. The energy is found in the bonds of the phosphates. So the more phosphates you have, the more energy you carry. The less phosphates, the less energy you carry. So ATP is an energy form that is found in your body, and it moves around, and whenever you want energy, you just take the energy out of that ATP. So with that in mind, look at this picture. You see all these actions that are happening? The woman is eating, the baby is sleeping, they're talking, the one is studying, one is running, one is doing gym. All of these cannot happen if you're not doing cell respiration. Like even eating, you need to move your hands, right? And when you're eating, you're breaking down glucose. The baby, for instance, he's sleeping, but who says that everything stops? No, his heart's still bumping, his lung is still moving, so that means they still need energy in order to do their functions. And so goes with the other pictures. So through all of these processes, you are actually going through cell respiration. And this is the reason why you are always breathing oxygen and you are always releasing CO2 all the time. Of course, there are like seconds when you can go towards anaerobic, which that means you're not breathing oxygen. But some people can do that for 30 seconds under the water. Some people can do 4 hours, but that's another story, right? A lot of training. But we can go for maybe a minute without breathing oxygen, but then we finish and we're like, we want to breathe all back. And that's because we are killing the amount of energy that we're not using anymore. So cell respiration happens on a daily basis, every second. As long as you're breathing oxygen and releasing CO2, cell respiration is happening. So in cell respiration, as I was saying, depending if you have the oxygen or not, so we have 2 ways of sections. We have the anaerobic, and you can see the word AN here, which means no. And aerobic here means oxygen. So that's what this sigla means, no oxygen. So in this case, anaerobic means you are doing without oxygen. Aerobic here, you can see there's no N, right? It has none of that. So that means you are doing with oxygen. So there's 2 ways. Or you're doing with oxygen, or you're doing without the oxygen. If you're doing without the oxygen, you are producing a small yield of ATP. Usually it's maximum like a 2 ATP. That's not much. And we're saying this according to glucose, okay? Now, if you are using the oxygen, that means you're fully breaking down glucose. So that means you are producing a large amount, and usually it's about 38 ATPs. So look at the difference. So you can see in the process that all of this is about breaking glucose. Like how much energy can you get out of the glucose? Let me show you exactly what I mean by that. So if you do remember, right, glucose looks like a hexagon like this, and then it has the CH2OH here, right? It has the oxygen, it has carbons, and it has hydrogens and OH. And you can continue, right, with all these guys, and continue here, and the H here, and then you have H here, right? You also have it here, and so forth. So I'm not going to draw the rest, but you just get my point. This is a glucose, as you can see. This is a glucose molecule. Now, think of glucose as the food that you just ate. That means you just took a nice hamburger and you digested in your stomach, and now that glucose is going through your blood, right? So let's say I have here, let's put it in red. Here's your blood vessels, right? That put in red is in blood vessel. And I'm gonna put green here as dots, and these dots are glucose, right? So glucose is in your blood vessel and it's moving, right? Because it left the stomach, it left the intestine, now it's in your blood. Now, the blood is transporting glucose around your body. Now, why is he doing this? Because he wants to give that glucose to all the cells in your body. So that means all the cells in your body need glucose to function. Now, when I say this, all your cells need glucose to function, it's because glucose has the energy needed for them to function. Where is that energy found in the glucose? It is literally on the bonds here. You see, every single bond that you see that I'm highlighting in yellow has energy. That's the potential of the bonds between them, all of these bonds. So every time you are breaking one of these bonds, so if you decide by any chance to break, for instance, if we break this bond between these two carbons, every time you break a bond with these two carbons, energy is released, like energy comes out of it. That's what he's doing. So it's like spa, and then you just see the energy come out of that bond. When the energy come out of that bond, then what happened is you have phosphates, right? Inorganic phosphates just floating. Think of them just floating around freely. What do? These phosphates come and pick up the energy, and now, voila, that phosphate is energized. He has energy now. Now, because he has that energy, that phosphate attaches to another ADP, right? So when they come together, right, these two, they come together, what you get? ATP. Oops, here it doesn't show ATP. So that means every time I am breaking a bond, right, like, oops, great. I'm breaking a bond, what you forming? An ATP. You break another bond here, you're forming an ATP. Break another bond here, you're forming ATP. And that's what you keep doing, is breaking these bonds. So every time you break a bond of glucose, eventually it will turn into ATP. So with that in mind, imagine here's the blood vessel, and next to it you have these cells, and all these cells are saying, hey dude, we need energy to function, to do our metabolism, to do whatever we need to do. So that glucose, what does he have to do? He has to leave the blood vessels, right, and moves inside the cell. So when they're moving inside of the cells, this is the moment inside the cell, the glucose that you find inside the cells, they are going to go through cell respiration. What does that mean? That means now the glucose inside of the cell are going to be broken down. So all these breaking downs that I showed you here, right? All these breaking downs, where does it happen? It happens inside of the cell. So the cell here, right? This guy here, what is he doing? He's breaking all the bonds of the glucose and sucking out energy from that bonds. In this way, he will use those energies to open channels, to make proteins, to create enzymes, to make things move or chemical reactions happening. That's why the cell needs glucose. It's just to suck out the energy out of the bonds. So that's why we eat food, because every time we break food, we are breaking those bonds and we're releasing that energy of those bonds in order to use it for our body. So that is the purpose of cell respiration. But to do that, not only do we need to get the glucose into the cells to do through this process inside of the cell. Let me raise this to give that space here. So inside of the cell now, imagine you are inside of the cell. So I have here a huge cell, and glucose finally get inside of the cell. So glucose inside of the cell, you need basically the cytoplasm of the cell as well as a very important organelle. So when it comes to cell respiration, you need the mitochondria. So here we have the mitochondria with its zigzags right here, right? So mitochondria is very important for cell respiration. Why? Because when it comes to breaking down glucose, glucose will first be broken down into two pieces. Now, just as a reminder, we know the glucose is made of 6 right? C6H12O6. So in the beginning, glucose is broken down. Now think of glucose as a chain of 6 carbons, right? 1, 2, 3, 4, 6, right? Because there's 6 carbons. So imagine this. So glucose eventually is broken down into 3 chains. It's broken down to two chains of 3 carbons, right? So when you break bonds and you make just now, imagine it went from a 6 carbon chain to 3, they changed their name. So now it's known as pyruvate. All right, so it's pyruvate. So we have 2 pyruvates right here. Now, when you do that, right, your glucose you're breaking, when you break that bonds, you are forming some NAD ATP and some NADH, which this will be more explained if you're an HL, which is later on. But this is carrying some energized hydrogen and some energized phosphate. So you kind of got some of the energy of the glucose. Now, these pyruvates will also move into the mitochondria. And here he's going to go through something called a link reaction, a Krebs cycle, and the ETC. All of this happens inside the mitochondria. But for SL students, you don't have to know that detail about them. You just need to know that they go through that process. And when they go through these 3 processes, eventually it produces about 38 ATP. So what happened through this entire process? The glucose was broken down, all the bones was broken down. And out of that, you got yourself the 38 ATP. So not only does in the cytoplasm, right, of the cell, glucose is broken down, but most of the glucose is broken down inside the mitochondria. This is why we say mitochondria is the powerhouse, ATP powerhouse. It produces ATP, right? And it's because that's where the mitochondria finally breaks down the entire glucose. And this is definitely the moment that you absorb all that energy coming out of the bonds of the glucose and you produce that 38 ATP. So every glucose makes a 38 ATP. And in this case, also, in order to break all these, to go through these 3 processes right down here, there's one element that is very important that it must be inside the mitochondria. It's oxygen. See, without oxygen, none of these 3 processes can happen. That's why oxygen must be inside the mitochondria to make that 38 ATP. Otherwise, it won't work. Now, of course, there's names to each one of this process, and we get to it. And now you got an overview of what cell respiration is about. It's all about breaking that glucose to make that ATP. So here is a picture of the mitochondrial structure, which you have to know. So eventually you will see that it has different parts, right? It has here, as you can see, I'm going to use the highlighter, which is better. This part right here is the outer membrane, and then that line here is the inner membrane. Now, the space between the outer and the inner membrane, which is right here, is the intermembrane, which the word inter means between. Now, the blue area here is the matrix. Now, you see the foldings that are happening here? These foldings right here is the cristae. Now, believe it or not, there is ribosomes inside mitochondria, and there's granules, which basically hold some sugars in there. And you can see some ATP synthase particles, which that means they produce ATP here. Okay, and they usually are found in the cristae. Now, this is a drawing. Now, if you actually had to draw this, let me erase this. Like if the teacher asked you to draw the mitochondria, labeling it for this process. So let me go with this. So we have here oval shape. So this will be the outer membrane. And then you will have the zigzag for the inside. Now that zigzag right here, that's the inner membrane. And then the space between them is called inter because the word inter means between. So intermembrane. And the space inside of the mitochondria is known as matrix. Now you see these little foldings that are happening here? These little foldings here, they are actually called cristae. Okay? And this is what you need to do, how to draw basically a mitochondria. Now, I told you that there is processes for glucose, right? So glucose, let's say it's out here. Glucose first goes through something called glycolysis, which is the first step of cell respiration. Then it goes through link reaction, and then it goes through the Krebs, and you can see they both, link and Krebs, they're both happening. So this is the second reaction, this is the third reaction. They're both happening at the matrix. And then you have the fourth reaction, which is happening here, which is the ETC, which is the electron transport chain, which happens in the cristae. So basically, 1, it happens in the cytoplasm. 2 and 3, these ones are happening at the matrix. You can say the matrix of the mitochondria. And number 4, it's happening at the cristae. That means it's happening at the inner membrane, right? Because the cristae is part of the inner membrane. Or you can also say it inner membrane here, but if you want to go more specific, cristae is the right answer. So, as you can see, there are 4 stages, right? The first stage is glycolysis, the link, the Krebs, and the ETC. You do not need to know what exactly happened each one as an SL, but as an HL student, once you get to 8.2 and 8.3, when it comes to photosynthesis respiration, cell respiration, you have to know more in details. But for SL, this is the maximum you need to know. And you need to know that oxygen is found here in the matrix. And oxygen is the very important key acceptor, okay? He basically is the reason why you can go through the link reaction, the Krebs cycle, and the ETC. Without the oxygen, none of these 3 can happen. Glycolysis can happen, but the link, the Krebs, and the ETC, they cannot happen without the oxygen. So you have to have the oxygen in the mitochondria. So moving with that in mind, here is a picture of the micrograph, which you need to know how to label the micrograph. And you can see here that the matrix is usually the space inside here. And then you have these foldings. You can see with the black lines here. These are basically the cristae. And then there's 3 lines right here. There's a line black here, there's a black here, and in between. That's how you find the outer, the inner, and the intermembrane. So make sure you get very well acquaintance with this picture of a mitochondria, because they might put that mitochondria out for you to see and say, hey, is this a mitochondria? So you really need to be well in identifying a mitochondria through a micrograph. So I just explained generally what was happening, right? So there is 4 stages. You have glycolysis, the link reaction, the Krebs cycle, and the chemiosmosis, which chemiosmosis basically means the electron transport chain, which is written here, the ETC. Now, what's happening on each one of them? The glycolysis, if you notice, it's the purple part here. It is happening in the cytoplasm. And this is the point where glucose is broken down to pyruvate. And here you only produce 2 ATP. Now, if you do have oxygen inside the mitochondria, then you're going to go through the link reaction, you're going to go through the Krebs cycle, and you're going to go through the electron transport chain. And in total, you're going to make a lot more ATP. So if you add these 3 together, you are making about 36 to 38 ATPs. That's what usually is that much. So with that in mind, this is just showing a general scoop that we have 4 reactions happening. Now, the word chemiosmosis, don't worry so much, that's for the HL. But for you, if you only put electron transport chain, you're good, okay? So these are the 4 sections to make the entire cell respiration. It all starts with glycolysis and it ends with the ETC. And glucose is broken down, and eventually in the end, you're making a certain amount of ATP. So for SL, you need to know their locations. So glycolysis happens in the cytoplasm, the Krebs cycle happens in the matrix, and the electron transport chain happens in the cristae. Do know their locations, because that's something that they might ask as a multiple choice question. So know their location. Now, let's talk about glycolysis, because this one you need to know a little bit more. Now, I already mentioned that it was a glucose molecule breaks into 2 of 3 carbons, which is known as pyruvate, right? So in the beginning, you have glucose, and the output is a pyruvate and 2 waters, 2 ATP, 2 NADHs. You do not need to know how these materials come out, but you need to know what's in and what's coming out of it. So what's really coming out of it is glucose, right? Starting in, and then what's coming out of it will be 2 pyruvates, right? Not only would you have the pyruvates come out, you're also going to produce 2 ATPs, and you're going to produce 2 NADH, which again, the hydrogen has energy because the hydrogen is coming from the glucose, right? Of course, you have water and more ATP coming out of it, but you need to know glycolysis, the reaction, what happens. And again, it happens in the cytoplasm. So this is the maximum that you need to know for glycolysis. Know that glucose turns into 2 pyruvates, and there's 2 ATP coming out of it, as well as 2 NADH. So here's the process of glycolysis a little bit more in detail. Of course, there is something called, like in the beginning of glucose, you are adding 2 ATPs because you want to put some energy to break the glucose into 2 pieces, right? Because you want to get those 2 pieces. But when you break those pieces, you release energy. So on that energy, you're producing 4 ATPs and 2 NADH. So at the total, if we talk about the net yield, right? Imagine you used 2 ATPs. 2 ATPs were used in the beginning, right? But then you form 2 ATPs. If you do the total, right, minus plus, in the end, what do you have? You have a net yield, which a net yield means a total yield. So overall, you made 2 ATPs during glycolysis, plus the pyruvates. So this is the only thing you need to know. Now, the NADH, which it is more explained for the HL students, it just means that the hydrogen, this hydrogen here, it's coming from the glucose, and this hydrogen has energy. So what is NAD doing? It's a carrier. He carries energies, he carries hydrogen as energies. So NAD just comes here and picks up that hydrogen that has energy and then transfer to the other part to be converted to ATP. But you don't need to know in that detail. So in the overall process here, this is showing all the energy that is produced. We can see that from one glucose, when it goes through glycolysis, and then it goes through the link reaction and the Krebs cycle, and then it goes through the ETC, overall, what you need to know is that in one glucose, for the good news of the SL students, you just need to know that one glucose will eventually make 30 ATP. Do you need to know in between the things here that are happening? No, but if you're an HL student, if you go to topic 8, you will know in more details how it's happened. But for the SL students, you just need to know that one glucose makes 38. So it goes back to the process. Now you're going to see here, but wait, this does not count like 38. Well, because again, here is not showing. It's usually 32, sometimes can be 32, sometimes can be 33, sometimes can be 34. So usually in books, you will see that it's written between 36 to 38 ATP. Okay, but this is just a recap. It's aerobic, and when I say aerobic, that means there is oxygen in the mitochondria, so you do have glycolysis, and then the pyruvate that broken down glucose enters and does the link, and this is the Krebs, it does the electron transport chain, and in the end, you're creating that 38 ATP. Now, what if you don't have oxygen? If you don't have oxygen, then your direction is not towards aerobic respiration, right? You're not making that 34 or more. That means you're not making that 30 ATP that I just talked, the whole thing that I just talked right now. If you don't have oxygen, you're not going to go this. Which way are you going to go? You go the other direction. So glycolysis that is happening right here, okay, that happens in the cytoplasm happens no matter what. With or without oxygen, glycolysis happens in the cytoplasm, and it happens. It doesn't need oxygen or at all. So you do break down glucose into 2 pyruvates naturally with or without the oxygen, and you do produce 2 ATP no matter what. It's only after you break down glucose to pyruvate, then you have to ask the question. So now you have 2 pyruvates, and the 2 pyruvates says, do we have oxygen? Oh, we do. Awesome. Then we go in this direction and you do the aerobic respiration, which includes the Krebs, the ETC, the link, right? And you eventually produce those 38 ATP if you have the oxygen. Now, if you don't, so the pyruvate goes says, do we have oxygen? And the body says, hey, we do not have oxygen. Oh, shoot. Then we're not going to go towards the aerobic anymore. We're going to go towards fermentation, which is known as anaerobic, which means there is no oxygen. So what happened is, now the pyruvate will not continue to be broken down. Pyruvate will turn into 2 options, depending on the organism. If you are a yeast or a plant, right, then the pyruvate will turn into alcohol and CO2 as a release product. And if it's in humans like us, right, the 2 pyruvates, it will turn into lactate, and that means you're going to have lactic acid is going to cause you to get tired very fast. Now, these two are known as anaerobic, which means fermentation, which means there's no oxygen, which means pyruvate will turn to stuff. Now, the most important thing is for you to look in this entire picture is the amount of energy that is created on the two sections, right? You look, if you do have oxygen, you have producing more than 34 ATP, sometimes 38, right? But if you do not have oxygen, do you know how much ATP you're producing here? Look, only 2 ATP. So in fermentation, you're barely producing energy. So without oxygen, you're not getting enough energy. That's why we breathe oxygen to get that energy. If we don't breathe, we get weak. So with that in mind, just to be clear, just to recap again, glucose going to turn into pyruvic acid. Great. Right now the pyruvic acid will ask, do we have oxygen or don't? Somehow we don't have oxygen. So if we don't have oxygen, that means we can't completely break down, right? Pyruvate. We cannot break it. Pyruvic acid and pyruvate is the same thing. You can't break it down. If you can't break down to the Krebs cycle, link reaction and stuff, then you're going to turn the pyruvic acid into lactic acid. So in total, you just made 2 ATP. Now, where do we have lactic acid? Lactic acid is every time you're not breathing oxygen. So for example, when you're doing those weightlifting where you hold your breath, or when you go under the water without breathing, so you're holding your breath for about 30 seconds or 1 minute, or when you're running for so long and your lungs is not getting enough oxygen because your lungs is so tired, so there's less oxygen in your body, right? Because this has happened whenever you exercise. Your lungs with time start to get tired. And when your lung gets tired, that means he's getting less oxygen in. So if less oxygen in, most likely you're going to start going towards lactic acid. So when you have too much lactic acid built up in your body, that's when your body starts getting tired. So going back here to the absence of oxygen, it depends on the organism. So for example, in yeast, you can make bread and alcohol, which that means in these all processes, you're making carbon dioxide and ethanol. If you're a bacteria, you can make yogurt and cheese with that, okay? Which you're producing lactic acid. Again, all of this is because there is no oxygen, and that means they're going through fermentation. It can be clearly seen in this picture by BioNinja. So here you see glucose turn into pyruvate, that it's given. We already know that this is happening no matter what. Glycolysis is happening in the cytoplasm. But then pyruvate, notice that there is no oxygen, right? No oxygen. Here, no. So that means depends on the animal. If you are doing with this two bacteria right here. So if you put a pyruvate with this bacteria, you're going to produce lactic acid, which this is how you make soy sauce. If you put pyruvate with lactobacillus, but without oxygen, remember, it will make lactic acid and it will produce cheese and yogurt. Now, if you put the pyruvate with the Saccharomyces, which is basically a yeast, right? This is a yeast. You can create beer with ethanol and CO2. You can produce ethanol such as the wine. And you can also produce carbon dioxide, which increases what makes the bread becomes bigger and bigger. Like it makes the bread volume build up. So all of these products that you see here in the bottom, all of these products are made during fermentation. They're basically fermented food. That means there is no oxygen. And that means pyruvate will turn into one of these products. So you do need to know this really well. Do you have to know the full name of the yeast and bacteria? No, you do not need to know. But you do need to know that pyruvate will turn into ethanol plus CO2 when you're putting in a yeast, right? You need to know that. And you can say that you can make wine, breads, whatever it is. You can also say that pyruvate turns into lactic acid in bacteria as well as human. And that can produce foods such as soy sauce and cheese. So just remember that this is because there is no oxygen whatsoever. Now, since we're talking about lactic acid, let's talk about us. Like, we do need to exercise, and when we do, we're using muscles, and muscles are using a lot of ATP. So sometimes when we're doing a very high intensity exercise, like we're running, we're sprinting, or such thing, it's going to reach a point where we're going to need a lot of oxygen, because you don't want to have a lot of energy for your muscles to do that fast running. But eventually what's going to happen is that it's going to slow down. But for you to get a lot of that energy, right, not only do you need oxygen, but you need to break down glucose to make the ATP, right? Because again, if you want the muscle to move, you need ATP. But for you to get ATP, you need glucose and oxygen. So all of the three, ATP, glucose, and oxygen, they have to be in large amount to allow you to do a high intensity. Once again, you need oxygen, glucose, and eventually ATP production in order to do a very high intensity exercise. So what happens if you don't have enough oxygen? That means you're not being able to break down glucose enough into the molecules that you want. So that means you're not producing enough ATP. So what happens to glucose? Instead of going to make a lot of ATP, glucose starts to become, the pyruvate, which it does this anyways, and then it starts going through lactic acid, which is right here. So pyruvate will turn into this compound called lactic acid. Now, lactic acid, the more lactic acid you have, the more your muscles will get tired. So increasing lactic acid gets the muscle tired. So that's why as you exercise, once you get to the 20 minutes, the 30 minutes, the production of lactic acid is starting to rise. And that's why you start to get tired at the 30 minutes and the 40 minutes, because now you have so much lactic acid in your body. Now, some people, they have better, I would say they have fitness, and that means their lungs are much stronger. So that means they can breathe more oxygen. So for a long time, they do not produce lactic acid. They start producing lactic acid maybe starting at the 40 minutes. That's why they only get tired after 1 hour running. But there's a lot of people, especially beginners. If they're beginners, that means their lung is so weak, right? The lung is so freaking weak that it's not taking enough oxygen. And then suddenly you're telling the lung, hey, dude, I need you to work faster. I need you to bring more oxygen because I'm doing exercise. So I need a lot more energy. So the lung is working so hard that by the 10th minute, he's like, dude, I cannot take enough oxygen anymore. I'm tired. So what happens to pyruvate? Pyruvate start turning into lactic acid. So that means by the 12 minutes or 13 minutes, you're starting to get tired and you cannot continue. So this is why we say, build your lungs, build your muscles, build your heart. The more you build them by training, the more you can spend a lot of doing exercise for a lot of time, because the lung will take more oxygen in. And if you have more oxygen, that means less chances of you doing lactic acid. That's the whole point. So logically, this is how it goes. If you're fit, that means it will take a longer time for you to start producing lactic acid. If you are not fit, that means you're probably going to be going through producing lactic acid much faster. Okay? So in this whole process of, breaking down glucose in order to get energy, you have to remember that we can use carbs, lipids, and proteins to do that. But all of this needs oxygen, right? So sometimes if you don't have oxygen, you start getting cramps on your body because your body is producing so much lactic acid. So that's when you stop exercise, you sit down and you go and you try to get that breathing back. And when you get that oxygen back, then you can go back to pyruvate from lactic acid and back to go through aerobic and produce more ATP. So let me explain this more clearly how it works, because most of the thing I was saying is just talking, right? So just to get it clear, you always have glucose converted to pyruvate, right? So it's all good. Pyruvate, up to here, you have 2 ATP produced. Now, let's just say you start to exercise. If you have oxygen, great. You have 30 ATP coming in, and that means you are getting all that energy and you're pumped up, you're fit. But if you start to have no oxygen, why would you not have oxygen? Because somehow your lung got tired, right? That means you're not fit. It means that you do not have strong muscle, and you probably don't have enough blood vessels, and your heart is weakened, and you don't have enough lung tissues going on. So with that, the lung got tired, so it means less oxygen. When you start having less oxygen, pyruvate will start converting into lactic acid. So The more tired the lung gets, the more lactic acid you're doing. So what's happening to lactic acid? So lactic acid, right, is starting to rise. And when this happens, you get more tired. And basically it's the body telling you, you need to stop. You need to slow down because we don't have what? Oxygen. So what do? You stop. When you stop, you start to catch your breath again, right? And you start to get the oxygen. When you get the oxygen, this is what happens to lactic acid, right? Lactic acid converts back to pyruvate. And from pyruvate, it goes this direction and start producing ATP. That's why people, when they get tired, they stop, they take a 10-minute break, they get that oxygen back, their lungs are back to rest, everything is back to rest. Now they're back into aerobic, which that means they're back to getting oxygen. And now they go back to producing ATP. So then after 10 minutes of resting, they can go back and do more running, right? But if you don't rest and there's not enough oxygen, lactic acid will just keep building and building, right? It just keeps building and you get more and more tired to a point you might, out of exhaustion, faint. That's why lactic acid is like a guy to give you a warning. Hey, dude, we have so much lactic acid. Slow down. You need to breathe. But if you don't have much of that, then you're fine. So when it comes to athletes, so just to give you a little scoop of that, when it comes to athletes, let's say you have an athlete here and you have somebody that is sedentary. Again. So the athlete, he does exercise every day, so definitely he has a very He has a very strong lung, right? And he has a strong heart. So for the first 30 minutes, he's getting enough oxygen and great, he's making that 38 ATP, lovely. Breaking down the entire glucose all the way to make that 38 ATP, lovely. And then around the 40 minutes, the oxygen is dropping. He doesn't have. So what happens now? Now he's producing lactic acid. And eventually by the 50 minutes, he's tired. He's done. He can't run anymore. He stops, right? Because too much lactic acid caused it to go. The sedentary, they have weak lungs, right? And they have weak heart and less blood vessels and stuff. So what happened is they probably can do for the first 5 minutes. They have oxygen, great. They're breaking down glucose and they're making those ATP out of the glucose. However, around the 6 to the 8 minutes, what's happening to the oxygen is dropping. Why? Because the lung is tired. So now he starts to produce lactic acid and it's starting to rise. So by the time he gets to 10 minutes, what happened? He's completely tired and he stops here. So that's the difference between somebody who is fit and somebody that is not fit. So can you become fit? Yes, you can. What do you have to do? This is what we call acclimatization, which means usually here, when the sedentary gets to 10 minutes, he's tired, he should push another 2 minutes of walk or running or jogging to start, forcing the lung tissue and the heart to pump more, to be stronger. And then maybe after 2-3 weeks, they can put another 2 minutes. So every time, what are you doing? You're increasing the minutes to make your body more used to it. And this way, in about maybe 2 to 3 months, the sedentary can become a fit person and start running actually for an entire hour. So that's why we always say it's okay to start an exercise with just 5 minutes and then build to 10 and then 15. But you got to start. So you can get away from being sedentary to athlete. It is up to you, but you got to be persistent. You got to do it every single other day if you want to. But as long as you always push, don't stop when you're tired. Go the extra 2 minutes, go the extra 5 minutes, because that will make your body work against the lactic acid. It will make your body build up his muscles, his tissues, everything. And this way you can run for a longer time with the oxygen instead of just quickly, dropping right here on 8 minutes without the oxygen. So lactic acid is more like a sign to tell you are tired or not. You're not getting enough oxygen. Now, here's another interesting thing is this graph. If you look at this graph, okay, you have here on the x-axis the exercise intensity. That means how hard the exercise is. Here, no exercise. Here, the exercise is really intense, very intense. This probably like be a HIIT workout. This probably could be running really fast, sprinting, right? And then here can be that you're not walking at all. So, and here around this part, it might be that you're just walking. Now, on the y-axis, we're talking about the percentage of energy, okay, from the fats and the carbs. That means, are you using the fat or the carbs? Because the pink here is the fat, right? And the carbs right here, basically is the blue. Now, remember the fats, there is no, when you're breaking down fat, there is no lactate. And here's, I want to show you how is that interesting. But if you break carbohydrates, because again, glucose is a carbohydrate, you do produce lactate production once you don't have enough oxygen. So let's look first at the fat right here, right? So the fat is the pink. When you're just walking, you actually are breaking down more fat. But when you start going from walking to start jogging and then to start sprinting, look what's happening to the fat. It means at this point, you're not breaking fat as much. So that means you're not using as much fat. So here's the interesting part. If you want to burn fat, what do you have to do? Walk, right? Look at carbohydrates on the other hand. Carbohydrates, when you're walking, you're barely breaking down carbohydrates, which that means you're barely producing any lactic acid. So when you're walking, not much carbs is going on, but then you start to jog. Now suddenly you're starting to use the carbs. So that means now you're starting to use lactic acid and you're starting to run and then you're sprinting. So what's happening now? You're using more carbohydrate, which that means you're producing more lactic acid. So now in high intensity, you do burn carbs, right? But you produce lactate. So here's the interesting part. If walking burns fat and running burns carb, and you want to lose weight, what do? And that's the logic here. If you want to burn fat, go walking, do not run. Because if you're running, it means that you are burning carbs. And here's the most interesting part is that when you are running, you get tired faster. Why? Because you are producing lactate. But when you're walking, you don't get tired so fast. You can walk hours if you have to. Why? Because you're burning fat and there is no production of lactic acid. So you're not going to get tired. That's why we always tell people, if you want to lose weight, walk. But if you want to get fit, run. That's simple. You want to get fit, run. If you want to lose weight, walk. Or even better, if you have a treadmill and you incline, keep the treadmill inclined, right? You're walking really slow on the treadmill on incline, you're more likely to lose that weight. Okay? So this is just to show you like how there is the consumption of carbs and lipids and how much energy it gives. So fat will only give his energy to you when you're walking, and the carb will only give its energy when you do intense exercise. Now, that was it. Hope that gives a little bit of a light also if you want to work out in your body. Now, you also have this experiment called the respirometer, which we're trying to measure the respiration rate. That means how fast respiration is going. Is it going slow? Is it going too fast? So here is an equipment that you have to know, be able to explain, or if you spot in the multiple choice. Usually we'll have 2 test tubes, and you will have the same amount of a solution here, which is KOH. This is basically an alkali solution, and this is a solution that absorbs CO2. So you can have KOH on this side, and you have KOH on that side. Exactly the same amount. Now, what is the job of KOH? And it says here, right? It is the absorption of carbon dioxide. So he absorbs CO2. Now, then you have a gauze right here. This gauze is to separate the specimen, which the specimen has to be a living thing. It can be a worm, it can be a cricket, it can be a hamster, whatever it is. You're putting an animal or a plant, whatever it is in there. And then you have that gauze to separate from that. So they can actually ask you, what is the gauze about? And they're going to say, hey, the gauze is separating the animal. Imagine you just drop the animal into the KOH solution. That's ridiculous, right? So you have to have the gauze to separate the specimen from it. And then, let me use the highlighter in this case. Then you have the test tube completely blocked by these corks, right? Because you don't want oxygen in or out or CO2 in and out. But you do have a tube that is connecting them to this manometer right here, which basically contains a colorful liquid in which it moves according to the pressure, and it's connected to the other test tube. Now, what is this trying to do? So we have to talk about pressure a bit here, so maybe you understand what I mean by that. So inside of here, you got to think there's oxygen, and here there's oxygen. So they have the same amount of oxygen, the same amount of KOH solutions, okay? And they have a specimen here. Now, remember that there is pressure here, so that pressure is actually hitting in this part, and this pressure right here is gases are hitting this part. That's why you have a level up, right? The liquid is level up because the pressure is keeping there. Now, imagine the organism here is starting to breathe that oxygen. So what's happening to the amount of oxygen in that area? It is starting to drop. When that drops, that means the pressure is dropping this way, and that means this liquid will start moving up. So this is how you can see cell respiration is happening, because the organism is getting the oxygen. Here's a better picture. Let me explain this one about the steps for you. So we have here, it's the same scenario as this one, okay? It's just different equipment, but it's still the same kind of scenario. See, let's look at this. You see the KOH here? Well, we have here a CO2 absorbent, which can be KOH liquid, but in this case, it's just an absorbent. Now, it has to be the equal amount, right? This you have to put on the other side that is the same amount as the specimen and the absorbent. So you can see that you have the KOH solution, same here, same amount. You have the gauze right here, right? This gauze right here, what is it doing? It's separating the specimen from the absorbent. You can see there is, oops, let me erase this so you can see. You can see that there is the gauze right there, right? What is the gauze doing? It's separating the specimen from this. Then you have these tubes right here connected, right, with basically a liquid that is colorful, right? Now, this experiment again is to see the rate of cell respiration. So what's really happening? You have in this area, you have a certain amount of oxygen, exactly the same amount as oxygen is here. Because of that, there's also oxygen coming here, and the oxygen is putting pressure, so it stops them here. And the same amount of oxygen here is putting pressure, so they're both putting the same exact pressure, right? And they have the same amount of volume of gases. That means the same amount of gases are here, it's exactly the same amount of gases there. So everything is equal, and because they're equal, you can see it right here, the liquid is equal to. So equal pressure on both sides, equal volume, everything is done. However, then you start the experiment. When you start the experiment, what is happening? This little animal here, what is he doing? He's taking in the oxygen. So when he's taking in the oxygen, what is he releasing? He's releasing CO2. Now, he's releasing CO2. Now, if you take off the oxygen and you put CO2, what happens to the pressure inside this thing? It stays the same, right? Because you're replacing one gas with another gas. So the pressure doesn't stay the same, and this will stay the same. It won't change because it's just replacing one gas with another gas. So there's no point in that. This is the reason why whenever the oxygen is coming here, that CO2 that is coming out of the animal, where is it going? It's going into this little guy right here. So it's being absorbed by this absorbent here. So it can be, if it's KOH liquid, then the liquid, okay, that K does not look like an H. All right, the KOH here is absorbing all the CO2. So think the logic here. Let me put this in the box. If you have a box here, if you have dots right here, right? And if I decided to take 4 dots out, that means I took 4 oxygen out. If I put 4 oxygen out and I put 4 carbon dioxide in, guess what? You have the same amount of gases, same pressure, nothing changed. But if I take the 4 oxygen out and those 4 carbon dioxides that was there, I take it out and I put it into an absorbent, what happened here? You notice there was less dots, right? And if I take another, let's say 4 oxygen here, and they convert them into carbon and I put it in the dots here, which means I'm putting here, guess what? Even less dots in that box. If it's less dot, which means less pressure, that means they're taking less volume in this, right? Like they're occupying less volume, which that means the volume is basically becoming bigger. But the point is less pressure. Now, if that is happening, what is happening to that pressure that was here? It's starting to decrease. Why? Simply because if there is less oxygen here, think about it, there's so much oxygen in this dot right here, right? But if I am taking slow bits by bits oxygen out, what is happening to these oxygens? These oxygens are starting to move here to fill up the place because, again, the animal is breathing that oxygen. So when this is happening, what's happening to the oxygen here? It's starting to reduce. Now, since there is no oxygen here to put the pressure, what is happening to the liquid? The liquid is starting to move up. So let me put in red, the liquid start going up like this because there's nothing to pressure. And if I keep taking more oxygen out, that means less oxygen is here. What happens to the liquid? The liquid start moving because there's no pressure on it, so it just keeps moving. If that is happening, what is happening on the other side? The liquid here start to disappear. It start to go down. So this is going down and this is going up. Now, this movement of liquid, you can measure the distance from here to here. So you saw, if I raise this, like make it more clear so you can see what I mean. So what I can do is I take a timer, I put 1 hour, I put it like, I want to see in 1 hour. And I start and I let him breathe. In 1 hour, he was here, right? And that distance. And then after an hour, he was here. What do? I measure the distance. So I took the distance that liquid traveled. Now, what is rate? It's basically the speed. So we know that is the distance over the time. So we have the time, which is 1 hour, right? We took an hour experiment, and then we measure the distance that this liquid went to go up. And we put the distance. So let's say it went 30 centimeters, whatever. So we basically divide 30 centimeters by that hour, and we get the rate. That means how fast the cell respiration was happening. So this is what this lab is about. It's just to see how fast this cricket is breathing the cell respiration. For that, we need these equipments. So the equipment such as the CO2 absorbent, because you don't want the CO2 to replace the oxygen, right? You want to keep it away. So you allow less pressure here. You have the manometers, which is basically seeing how the pressure is changing as the water is moving around because there's less oxygen causing pressure on it. We also have to put on this side an object that is exactly the same size as volume and mass as the CO2 absorbent and the specimen. And this has to be closed up, right? Because you don't want to mix the oxygen of the air with the oxygen inside. So this is a very good type of experiment to just measure their cell respiration. But we cannot use this as an IA because, again, we cannot play with animals. That's against the IB rules. But this is a respirometer, which we're just seeing how fast cell respiration is happening, okay? So here we have basically the steps written in words. You can pause this video and read it because I know some of you guys have a hard time writing in words, but it's very straightforward. So you get a living specimen, okay? You don't have to say if it's germ seed or anything. You put them enclosed, a sealed container, right? Then what you're basically doing is measuring how much carbon dioxide will be coming out of it, right? You can put a logger in there, a pro, just to check. You can have an alkali. In this case, an alkali is like the KOH, which absorbs CO2. Now, the oxygen will be consumed by this cricket or whatever the animal is, and you would see the change in pressure. So the change in pressure, we're talking about the change in pressure that is happening here. And you can detect, like I said, the YouTube manometer, right? This YouTube right here is the manometer. So this is where you can see the change in pressure. Why? Because the motor is moving up. Why? Because there's less oxygen. And once you say that, you can say what are the rates that it comes out. Now, there's many ways that you can play with this. You can play using temperature, water, light, age, activity levels. So, like, you can put a lamp here and put heat towards this animal and see how the animal is, if he's going to breathe faster or slower. You could present, for example, you can get an age. You can make 2 samples of these guys, right? 1 sample here, 1 here. Here you put a kid that is 5 years old, and here you put a kid that is 10 years old. Of course, you're not going to put the kid on the beaker, but I'm just giving an idea. And you check which one does cell respiration more faster, and so forth. So this is different ways that you can manipulate the respirometer. Why am I saying this? Because they might ask you, like, give me examples of how you can manipulate this experiment right here. Like, how can you manipulate? You can manipulate with temperature, you can manipulate with age, you can manipulate with activities. Like, what I mean by activities is you can make one cricket walking, the other one running. I know crickets don't walk, but I'm just giving you ideas, okay? So anyways, this is a respirometer. And this is, again, like I said, it's just to measure the aspiration rate. And this is doing by checking the pressure changes as the oxygen is being absorbed by the cricket. And the CO2 that is released is kept inside the CO2 absorbent. So this is a perfect way to do it. And that's pretty much. Here's a study guide. You can go through these questions and go back to the videos to find the answers. And that's it. Thank you.